Happy Monday. So a couple of announcements. You know that if you looked at the syllabus, there's an exam on this section of the course 2 weeks from today, and there will be a handout posted on BSpace about this. I hope all of you had a chance to download the study questions that I posted there last Friday, and I will be posting another list of study questions probably towards the end of this week on the material that we're covering this week. We're also going to be having I will be holding an exam review on Friday night, February 19th, 6 to 9 p.m. or 6 p.m. Let's just say 6 p.m. till whenever we're done. Okay, 6 p.m. till whenever we're done. And come with all your questions to that review, but of course you can come to office hours before that. Okay, so last week we talked about enzymes, and we spent a fair amount of time on that. We spent 3 lectures on that chapter. And somebody said, why are we spending so much time on enzymes? And the reason is that, one of the reasons is that enzymes really power all of the activities of the cell and of organisms. And so this week you'll see in some gory detail how enzymes play a critical role in fueling all of the processes that go on in biology. So we're going to talk this week about the material in Chapter 9, which is about cellular respiration, really understanding how it is that cells are able to either produce their own fuel, if they're plant cells, or take in fuel, if they're animal cells, and burn it and use it to make useful, to do useful work in the cell. And so what we're going to do today is we're going to talk about cellular respiration, sort of an introduction to this. And what I want to do is to focus on 3 topics. One is understanding what redox reactions are, oxidation reduction reactions. We have to understand this to understand how it is that cells use fuel to make ATP. We're going to talk about the electron transport chain a little bit today and how that works, sort of an overview of this process. And then we're going to talk about glycolysis, which is the first step in cellular respiration. All right, so first of all, life is work. You probably knew this. But when we talk about it in molecular biology, we're referring to the fact that living cells require energy from an outside source. So if it's an animal, animals will take in energy either by eating plants, like this fellow right here, or by eating organisms that eat plants, if they're carnivores. And if they're plant cells, they make the fuel directly by harvesting energy from sunlight. This is a slide from the book that sort of illustrates this concept. So energy comes into our ecosystem first in the form of sunlight here, where it's harvested by photosynthesis in plants. So plants will use input energy from sunlight together with carbon dioxide and water to produce sugars that are made here in plant cells, makes these organic molecules. These provide the fuel for respiration that occurs in mitochondria. This is going on both in plant and animal cells, right? So plant and animal cells have mitochondria that are burning the fuel, but just plant cells have the chloroplasts that are making sugars to begin with. And the end product of all of this, the respiration pathway, is the production of ATP molecules. And it's ATP that powers most cellular work. And so this is a really fascinating process. How is it that cells are able to start with fairly big organic molecules, sugars at least, and break them down and produce one type of molecule, ATP, and then harness it to do all sorts of different kinds of cellular work? So that's really what we want to talk about today, right here. All right, so catabolic pathways are we talked about catabolism, right? So this means that we're breaking down larger molecules, such as organic molecules, to produce smaller molecules. If we talked about anabolic pathways, we're talking about building up larger molecules from smaller building blocks. But if we're talking about respiration, we're talking about catabolic reactions, because we're thinking about the breakdown of organic molecules and the harvesting of that energy that occurs in the process, because this breakdown is exergonic. That means energy is released in the process of doing this. Now, that energy, that release of energy could just be released as heat, right? But cells actually want to harness it. They want to couple those breakdown reactions to the production of other molecules, ATP specifically, that can be used to do useful work. And this is done in 3 different ways. So fermentation is a process that involves partial degradation of sugars, and this is occurring in the absence of oxygen. Okay, and we're going to talk about that more later this week. We're not going to talk about fermentation today. Okay, but you should know that it's a partial degradation of sugars that occurs in the absence of oxygen. Anaerobic respiration is really what we're referring to usually when you hear the term cellular respiration, and your book talks about this, right? So cellular respiration, if you hear that, typically means that we're talking about aerobic respiration. If you see the word aerobic, you immediately know that it means oxygen is involved, right? And so that's exactly what this means. It's in the consumption of organic molecules together with oxygen to yield ATP. Now, it turns out that some kinds of cells can also do another kind of respiration that can occur when there isn't any oxygen around. And if there's no oxygen around, they can still burn fuel and produce energy through the process of anaerobic respiration. All right, so this consumes compounds other than oxygen to break down organic molecules. All right, and so how does this work? And so to understand how energy is harvested from food, ultimately, from sugars, we have to first talk a little bit about redox reactions, what those are and how they work. So if you see redox, this is just a combination of reduction and oxidation, right? And so this means that this involves the transfer of electrons that occurs during chemical reactions to release energy that is stored in these organic molecules. Ultimately, that energy is used to make ATP, but as you'll see, not initially, right? There's a number of steps that have to occur before we get to the production of ATP. Okay, so chemical reactions that transfer electrons between reactants are called oxidation reduction or redox reactions. These are always coupled, right? So this is why we use the term redox. Reduction and oxidation are coupled in the same reaction. During the oxidation, in the oxidation step, a substance is losing electrons, becoming oxidized. In reduction, it's gaining electrons or becoming reduced. And the reduction refers to the amount of positive charge that is being reduced or gained in this case. And the next slide, I think, shows just a very simple example for sodium chloride. And we can actually write this in an even simpler way, or an even more general way if we want to. Okay, and so this right here is called the reducing agent. Okay, and so this is the electron donor. The reducing agent is the electron donor in the reaction. And this is the oxidizing agent. And you got it, it's the electron acceptor. So you can see why oxidation and reduction always go together, because the electron transfer requires both a reducing agent to donate the electrons, and it requires an oxidizing agent to accept electrons, right, in this type of reaction. And we'll see this as we look at the process of how glucose, the major sugar that's burned during cellular respiration, how that's broken down, right? It involves a series of oxidation and reduction reactions, redox reactions. This just summarizes this. So the donor is the reducing agent, the electron acceptor is the oxidizing agent, and this is important to note. So this, when we looked at the sodium chloride example, or this general example here, this looks, with the way we draw this, the electron is being completely transferred in the reaction. But in some cases, redox reactions don't transfer electrons completely, but instead what they do is they change the electron sharing that occurs in covalent bonds. And a great example of this is the reaction that occurs between methane, which is just natural gas, right? It's what we burn in our, if you have a gas stove at home, this is burning methane. A lot of power plants burn methane, natural gas. And the reaction between methane and oxygen is a redox reaction in which there is not a complete transfer of electrons in the reaction, but it's nonetheless an oxidation reduction reaction. And we can look at it right here. So here's the natural gas, methane. This is the reducing agent in the reaction. And it's reacting with 2 moles of oxygen, the oxidizing agent, to form carbon dioxide and water with the release of energy. And so this is overall an exergonic reaction, right? We're releasing energy in the process, so the overall delta G of this reaction would be negative. And what you should notice here is, so that this is a figure from the book, so they've sort of drawn out the way electrons are considered to be shared in these bonds. And in methane, we have tetrahedral organization around this carbon of these 4 hydrogens in which electrons are shared equivalently in these bonds. And here's the oxygen that it's reacting with. This is a very strong oxidizing reagent, and the reason is that oxygen is very electronegative. That means it attracts electrons very strongly to it. And so you can see here that in this reaction, once carbon dioxide is produced, these electrons are held much more strongly towards the oxygen in each bond, right? Because oxygen is very electronegative. And so that is a process that allows electrons to be partially transferred, in this case, by moving into these bonds where the organization of electrons in the bond is not equivalent, but is in fact closely centered around the oxygen because it's so electronegative. So in general, if you'll see that in a lot of these redox reactions that we're talking about, we talk about oxygen being ultimately the potent oxidizing agent because of its electronegativity. All right, so this is now looking at an example that's a little more relevant to what we're talking about in biology, which is the fuel, which is glucose here, becoming oxidized to carbon dioxide and producing energy and water in the process, right? And so this is the reaction that we're going to be considering. But as you'll see, it's not a reaction that is occurring in one step, like is drawn right here, but is in fact broken down into a number of individual steps by enzymes in cells. And the reason for that is that it wouldn't be very useful to do this reaction all at once, because it would be very difficult for cells to actually harvest that energy in a productive way. Right, and I wanted to tell you one other thing while we're on this slide, which is that, as you can imagine, this is a very exergonic reaction, and the delta G for this is 686 kcals per mole. So this is a very exergonic reaction. So glucose is around, there's oxygen around. Why doesn't this just spontaneously combust? Why doesn't glucose just immediately react with oxygen? It's very exergonic. Activation energy, exactly, right? So what we talked about last week. So to do this, even though it's a very favorable reaction, there's an energetic barrier to that reaction occurring. And that's what enzymes do, right? They reduce that activation barrier so that it's easier for this reaction to occur. Even though it's a spontaneous reaction, and even though it's an energetically favorable reaction or exergonic reaction, it still has an activation barrier to get over. Okay, so this slide here really discusses the fact that in cellular respiration, glucose or other, and I should have said this earlier, but I should say that we're focusing on glucose, but you should know that other kinds of organic molecules can also be broken down for fuel. We'll talk about that in a later lecture this week, okay? But so in cellular respiration, these starting materials are broken down not at once, but in a series of steps, all right? And so you remember that last week we talked about enzyme cofactors, and one of them that we mentioned was the cofactor NAD, nicotinamide adenine dinucleotide, NAD. And NAD will come up again and again when we talk about these reactions because it's a very common cofactor that's used to shuttle electrons in redox reactions that are occurring during respiration. So they're typically what happens is that electrons from organic compounds are first transferred to NAD, which is NAD+, it's positively charged. And so it's an electron acceptor, right? It's positively charged, accepts electrons, functions as an oxidizing agent during respiration. And then each of the resulting molecules, which is a reduced form of NAD+, NADH, represents stored energy that can then be tapped to make ATP. All right, and so this is just indicating that if we start with a bond in a sugar, this might be a part of glucose, for example, and we have NAD+ around, enzymes such as a dehydrogenase enzyme, the ACE, again, remember we talked about ACE meaning that this is an enzyme, so you immediately know this is an enzyme, will catalyze this reaction and catalyze the transfer of electrons from the reducing agent to the to the oxidizing agent, the electron acceptor here. So I wanted to show this in a little bit more detail. So here is the nicotinamide adenine dinucleotide, and just take a look at this molecule for a moment. So you can notice that it's definitely a dinucleotide. Here's the ring structure. You can see it repeated, one unit here, and here's the adenine base. So this just looks like adenosine right here, right? This part with the here's the phosphate, and notice that there's a 5' to 5' phosphate linkage. So that's not a linkage that occurs in DNA or RNA typically, right? That's a 3' to 5' linkage in DNA or RNA, but here it's a 5' to 5' linkage. And then up here is, it's also a base, but it's not a base that's found in DNA or RNA. It's nicotinamide, here shown in the oxidized form. So here's the positive charge on this base. Here's the glycosidic bond right there. And this is, again, as we said, a very common agent that's used in cellular respiration to transfer electrons. What it does is if we have we have NAD+, and we have 2 hydrogens shown here, and then there's a mistake up here. I want you to fix this little error that I made when I was making this slide. This plus should be here, right? So we have 2 electrons and 2 protons that are being transferred in the reaction by this dehydrogenase enzyme, leading to reduction of NAD+, if it's going from left to right, and leading to the NADH form of this cofactor, the reduced form over here. And what you can see is that 2 electrons plus a proton are being transferred here, and then with release of a proton over here. So please also fix that, so that plus should be right there. So let's see. So I wanted to point out that I think we said this earlier, but most electrons that are removed from fuel molecules, food molecules, are transferred initially to NAD+. Okay, so very important cofactor of metabolism, and we have to have it. Yeah, question? Say again, is that? Well, it's, again, this is this is a situation where this can go back and forth, right? And so, and it's enzyme catalyzed, so it's a cofactor reaction. So it can be energetically favorable to go either direction depending on what the enzyme is and what the reaction is that's occurring. Okay, but typically when we're moving in cellular respiration, we're moving towards producing NADH, because this is building up to allow us to make ATP, and you'll see that when we talk about oxidative phosphorylation. Okay? All right, so NADH passes electrons into what's called the electron transport chain. And we're going to talk about this in much more detail later this week. But the important thing here is to know that this is not an uncontrolled reaction. So the electron transport chain, it really is a chain. It's a chain of reactions that passes electrons along in a series of steps so that we don't just get one explosive reaction of glucose with oxygen, which would not be very useful, but in fact allows cells to harvest energy along the way. Oxygen pulls electrons effectively down this chain in an energy-yielding tumble. So what does that mean, energy-yielding tumble? That means that these reactions in general are exergonic, right? They're favorable reactions. We're going from a higher energy state to a lower energy state at the end. We're starting with molecules that have a lot of potential energy, and we're harvesting that energy as those molecules are broken down into simpler molecules and ultimately into CO2 and water. And this energy that is harvested is used to regenerate adenosine triphosphate, ATP. Right? So cells are constantly using ATP to carry out work. We talked about that a little bit when we looked at how cells function, transport of molecules across membranes, for example, movement of flagella and cilia. All of those things are energy requiring, burning ATP, and so cells have to have a way to regenerate ATP, and this is the process of respiration, harvesting this energy that's produced from breaking down more complex molecules. This is a slide from your book that just shows that sort of a comparison of what happens if we have an uncontrolled reaction that's very exergonic, right? So very favorable. This would just be the reaction between liquid hydrogen, for example, and oxygen. This is a reaction that is used to fuel rocket ships and the space shuttle and things like that, right? It's a very exergonic reaction, and when you ignite these guys, they react to form water, and it's explosive. So that means there's a very rapid reaction that releases heat and light. Uncontrolled reaction. So this isn't not very useful to cells. Instead, what cells do in respiration is they carry out this series of electron transfer reactions involving this cofactor to release energy in a controlled, stepwise fashion. And in the process, and we'll see how this works in more detail today and this week, ATP molecules are produced in the process. Okay, so I now want to sort of start off this discussion of glycolysis by showing you just a preview of the different steps in cellular respiration. And so we can think about cellular respiration having 3 distinct stages associated with it. First one is glycolysis. Glycolysis is the breakdown of glucose into 2 molecules of pyruvate, right? So this is the process that really starts off cellular respiration. And so once these pyruvate molecules are produced, they enter a pathway called the citric acid cycle, which leads to the complete breakdown of glucose. And once that occurs, the products of the citric acid cycle are then used to drive oxidative phosphorylation. And this is where ATP synthesis, for the most part, is occurring. Okay, so most of the actual production of ATP is occurring at this last step of cellular respiration right here. So I don't know, maybe I'll just, to sort of, again, as part of this overview, I just want to sort of put this pathway on the board, which is showing the way that electrons are traveling. Right, so electrons are traveling along a route that starts with glucose, then moves to NADH, then enter the electron transport chain, and finally the byproducts are CO2 and H2O. Okay, so let's look now. Let's think a little bit about where this is going on in the cell. These 3 stages of cellular respiration. So here we're looking at a cartoon of part of a cell, and probably you know that sort of what that kind of looks like, right? What is that? Mitochondria, right? Yeah, so that's a mitochondrion right there. And so this reaction, glycolysis, is occurring out here in the cytosol. Okay, so it's a cytosolic reaction. So the enzymes that we're going to talk about that catalyze the steps of glycolysis are all cytosolic enzymes. And here's the overall reaction. So overall what's happening is that glucose is being broken down into pyruvate. And please put a 2 there, okay? Please put a 2, because each mole of glucose is producing 2 moles or 2 molecules. 1 molecule of glucose is making 2 molecules of pyruvate. All right, so this is, that's sort of something, I don't know, it's left out in the book, but in this picture, but you should put that there. And in the process of this breakdown, electrons are being transferred to NAD+ to make NADH. And in addition, as you'll see, we're also making some molecules of ATP. And we're doing that through a reaction called substrate-level phosphorylation. We'll look at that in a little more detail in a couple minutes. Okay, so we're both producing NADH through electron transfer, and we're also actually making some ATP in the process. So this is all going on in the cytosol. And then, once pyruvate is produced, it enters the citric acid cycle, which is going on inside of the mitochondria. Okay, and we're going to talk about that a lot more on Wednesday, the citric acid cycle, where these enzymes are located. But know for now that this is a process that's happening in the mitochondrion itself. And that leads to the further breakdown of these pyruvate starting molecules and the production ultimately of CO2 and water. And in the process of the citric acid cycle, we're also transferring more electrons to NADH and also to another cofactor. We talked about the FAD plus briefly on last week, and this is also a cofactor that's used for electron transfer. So both of these are involved at this step. And we're also making ATP, again, by substrate level phosphorylation. All right? And then finally, oxidative phosphorylation occurs, in which this is where the electron transport chain is going on, and a process called chemiosmosis. We'll also talk a lot more about this later this week, which is where the vast majority of ATP is made in this process, oxidative phosphorylation. All right, so that sort of gives you a sense of where these reactions are actually happening. Let's see. So one thing I wanted to mention. So this, of course, this is showing you what's happening in a eukaryotic cell, but prokaryotic cells also do respiration, right? And so where are they carrying out the electron transport? Anybody know? In the cell membrane, the plasma membrane, right? So it's a membrane. These enzymes are membrane-bound enzymes, so they have to work inside of a membrane. And in prokaryotic organisms where there isn't mitochondria or mitochondrion, they carry this out in the plasma membrane. Okay, so as we said, so the process that makes most of the ATP in the cell is oxidative phosphorylation. And this is really a set of reactions that are powered by oxidation and reduction. We'll talk about that more on Wednesday. 90% of the ATP that's made during cellular respiration is a result of oxidative phosphorylation, so the vast majority. And then a smaller amount of ATP is formed during glycolysis and in the citric acid cycle by what we said was substrate level phosphorylation. So what is that, and what's the difference? And I want to just show you this slide here. This is, again, a figure from the book that illustrates substrate level phosphorylation. And so what's happening here is that an enzyme is transferring a phosphate group from a substrate molecule to a molecule of adenosine diphosphate, ADP, right? So this is what substrate level phosphorylation means. We have a substrate that's phosphorylated, and we're transferring a phosphate from that substrate to ADP to generate ATP in the process and a dephosphorylated product that's released from the enzyme. So in oxidative phosphorylation, what we're going to see is that the way that ATP is generated is that we have a phosphate group, an inorganic phosphate, being added to ADP to make ATP. So that's the difference. Oxidative phosphorylation, we're going to see inorganic phosphate used to regenerate ATP, but here in substrate level phosphorylation, we start off with a larger molecule. So this might be a sugar molecule or a sugar byproduct that's phosphorylated, whose phosphate is then transferred over to ADP to make ATP. Okay, so glycolysis is the process of harvesting chemical energy by oxidizing glucose to pyruvate, 2 molecules of pyruvate for each glucose that's broken down. This is a word that literally means sugar splitting, right? Glycolysis. And so we're breaking a 6-carbon sugar down into 2 3-carbon molecules, these pyruvate molecules. And it occurs in the cytoplasm, as we said, cytosol. And there are 2 major phases. There, in the first steps of glycolysis, we have what's called the energy investment phase. And as we'll see, we actually have to input 2 molecules of ATP initially to start the process of breakdown of glucose. So we have to invest energy initially, and then in the later steps of glycolysis, we have the energy payoff, where we actually have a net production of ATP as well as NADH that's produced in the process. Okay, plus we get these two molecules of pyruvate that are the starting material for the citric acid cycle. Okay. All right, so this is a figure from the book chapter that shows the sort of diagrams this energy investment phase. So we start off with glucose, and as we're going to see in the next few slides, we have to initially invest 2 molecules of ATP. So these are burned and used to phosphorylate products that occur from enzyme-catalyzed reactions in these first steps of glycolysis. So we burn 2 molecules of ATP during the energy investment stage. And then those phosphorylated molecules are substrates for additional enzymes that are involved in the energy payoff phase. And so in this step, we're going to see that we actually have a net production of ATP because we can produce 4 molecules of ATP. And this is through substrate level phosphorylation that's occurring here. 4 molecules of ATP produced in addition to 2 molecules of NADH as well. And then we have these 2 pyruvates that are made here. And so if we do the math, what we get is that we have to use 2 molecules of ATP, but we're making 4. So we have a net production of 2 ATPs overall in the reaction. And we also have a net production of 2 NADH molecules in the reaction. And then we have these pyruvates that are ready to enter the citric acid cycle as well. Okay, so what I want to do now is I want to go through the steps of glycolysis and just talk a little bit about what's happening at each step and look a little bit at the chemistry. And these, again, there's a nice 2-page spread in your book that goes through each step of glycolysis with these figures and discusses what's happening at each step. And so you should really spend some time studying that. Okay, I really want you to understand the principles. You don't have to memorize the chemical structures of each of the molecules that in the glycolytic pathway, but I do want you to understand the principles, what's happening. I want you to know that a molecule of glucose is being broken down into 2 molecules of pyruvate. I want you to understand the overall energetics of what's happening, and I want you to know something about the enzymes and what they're doing in the reaction. All right, so this is the first step of glycolysis in which glucose is entering the cell and getting phosphorylated by an enzyme that we've actually seen before in class, this enzyme called hexokinase. You might remember that when we talked about substrates binding to active sites, I showed you a picture of the hexokinase enzyme that has a cleft that fits the glucose substrate very nicely and closes down around it when glucose binds. And what this enzyme does is it catalyzes the addition of a phosphate onto at this position in the glucose ring, on the glucose molecule right here, to make glucose 6-phosphate. So this is the first reaction that occurs in the glycolytic pathway, and it requires consumption of 1 molecule of ATP to phosphorylate the glucose to glucose 6-phosphate. So we can think a little bit about why do cells do this, and there's a couple of reasons that we can think of. One is that once this phosphate is added onto the glucose molecule, phosphate is very highly charged, right? It's very negatively charged, and so this molecule, once phosphorylated, then becomes impermeable to it becomes unable to go across the cell membrane anymore, right? Because it's highly charged. Because the cell is generally impermeable to these kinds of very highly charged molecules. So it effectively traps this product in the cell so that it can go on further in the pathway. And also, having the phosphate there, turns out it makes this molecule much more chemically reactive for later steps in the pathway. So it has at least a couple of reasons why the cell would invest energy to phosphorylate glucose in the first step of glycolysis. All right, and so then this glucose 6-phosphate is then interacts with the second enzyme in the glycolytic pathway, phosphoglucoisomerase, and the job of this enzyme is to basically isomerize glucose 6-phosphate into fructose 6-phosphate, which is shown down here. And if you look carefully at the orientation of the hydroxyl groups around the ring of the sugar, you can see that there's an isomerization that's occurring, right? These are changing their orientation with respect to the position of the phosphate in this group in the ring here, the oxygen. All right, so we have fructose 6-phosphate, which then is enters the third, reacts with the third, in the third step of glycolysis, catalyzed by this enzyme here, phosphofructokinase. You'll see this abbreviated PFK frequently, phosphofructokinase. Again, you know, a lot of these enzymes in the glycolytic pathway have been highly studied. PFK has been studied for a long, long time, you know, decades. We know we have many crystal structures of this enzyme. And so we know a lot about how it carries out this reaction. What it's doing, again, is burning the second ATP in the investment stage of glycolysis to put another phosphate group onto fructose 6-phosphate to make fructose 1,6-bisphosphate, which is shown right here. Okay, so now we have two phosphates on this sugar. Having these two phosphates here basically makes the sugar much more reactive, and it's now ready to be split in half, which is the real lytic step, the glycolytic step of glycolysis, right, in the next step of the pathway. So turns out that this enzyme, PFK, is a very highly regulated enzyme. This is a step in the glycolytic pathway where cells decide whether or not they're going to proceed. And so we know that, for example, this enzyme is highly subject to the kinds of feedback inhibition that we talked about last week, where the end products of the reaction will bind to this enzyme and inhibit its activity, so that if there's a lot of glycolysis going on and we have a lot of production of the products of glycolysis, they can rebind this enzyme and slow down the activity so that we downregulate the glycolytic pathway under those conditions. Okay, so it's a key regulatory step in the glycolytic pathway right here. All right, so then we get to the step in glycolysis where the sugar is actually split. And this is being catalyzed by aldolase right here, making two products. One is called dihydroxyacetone phosphate, or you'll see it abbreviated DHAP right here, and also a molecule of glyceraldehyde 3-phosphate over here. Now, importantly, there's something kind of really interesting here. So there's another enzyme, the fifth enzyme in the glycolytic pathway called isomerase, which is an enzyme that catalyzes the interconversion of these two products of the aldolase reaction. Only this product, only glyceraldehyde 3-phosphate, is the product that can enter the rest of the glycolytic pathway. So we need this isomerase to convert dihydroxyacetone phosphate, DHAP, into glyceraldehyde 3-phosphate so that it can be used also in the reaction. So even though this is, you see these arrows going back and forth, so this is a potentially a reaction that could be in equilibrium, it actually never reaches equilibrium, right? Because in glycolysis, as soon as this is made, it's consumed in the next reaction, catalyzed by the next enzyme. And so that drives this equilibrium pretty far to the right, and this isomerase is basically going mostly in this direction, making glyceraldehyde 3-phosphate that's then entering the rest of the glycolytic pathway. All right, so we talked about the energy investment stages of glycolysis, and now here's where we start to get the payoff. All right, starting in step 6. So we have this enzyme here, triose phosphate dehydrogenase, which is taking glyceraldehyde 3-phosphate and converting it into 1,3-bis-phosphoglycerate down here. In the process, we are actually producing 2 molecules of NADH from 2 molecules of NAD+ by the transfer of electrons. Okay, so there's an electron transfer reaction happening here, and there's also an input of inorganic phosphate. Let's see. Right, so this is overall, this is a very exergonic reaction, very favorable reaction right here. Okay, so energy is being harvested. Okay, and then in the next reaction, so in step 7 of glycolysis, now we're seeing the first case of substrate level phosphorylation that we talked about, right? Because here we have the 1,3-bis-phosphoglycerate reacting to form 3-phosphoglycerate, and this is catalyzed by phosphoglycerol kinase, this 7th enzyme in the pathway, and in the process, we have 2 molecules of ADP being converted to ATP, right? So we're producing 2 molecules of ATP in this reaction. Substrate level phosphorylation, because this is effectively the phosphorylated substrate whose phosphate is being transferred to ATP in the process. And it's 2 molecules of ATP because remember that at this point we have 2 molecules of the 1,3-bis-phosphoglycerate for every molecule of glucose that we started with that's going through this pathway. Yes? I think there's water involved in this too, right? Yeah, it's not shown here. Chemically, these are not very good pictures, but there's water involved in this reaction as well, okay? Okay, so these 3-phosphoglycerate molecules produced in the last reaction then react to form 2-phosphoglycerate via an enzyme called phosphoglyceromutase. These are wonderful enzyme names, by the way, aren't they? They're just great. Phosphoglyceromutase, and this basically, you can see the reaction here. We're basically relocating this remaining phosphate to a different position, here to here, preparing it for the next step in the pathway, step 9, catalyzed by an enzyme called enolase. And in this reaction, we can see that we have 2 waters coming off, and we're producing phosphoenol pyruvate right here. So we're almost to pyruvate, not quite. Phosphoenol pyruvate. This molecule has, again, very high potential energy. So the next reaction is very favorable. The last reaction of glycolysis, catalyzed by pyruvate kinase, which is taking phosphoenol pyruvate and producing pyruvate. And again, this is an energy-producing reaction because we take ADP and phosphorylate it to form ATP. Okay, so please review those reactions in the book and read the text. They have a very nice discussion of each step of glycolysis.